Hello everyone, my name is Mahendra Raut. Today I'm going to talk about Proteomics, the next generation tool to expand synthetic biology applications in a biofuel generation. So from this figure, you can broadly understand my research background. So basically I'm exploiting different microbial systems at proteomics level to understand their biology in great detail. And so today, particularly, I'm going to talk about lignocellulosic biofuel generation, and um, and I'm going to talk about how omics and synthetic biology tools to help to improve these bioprocesses. So, coming to the next slide, here is the brief introduction about me. I am a biochemical engineer. I did a PhD in biochemical engineering from the University of Sheffield, United Kingdom. I did my graduation and post-graduation in microbiology from Nagpur University, India. I have been practicing microbiology since 16, 17 years. Currently, I'm working as a postdoctoral research associate in microbiology at the University of Sheffield. I have also worked in India. I worked as a senior microbiologist at Haldiram International Food Limited. I also work as a research assistant at National Environmental Engineering Research Institute, India. So in my today's talk, I'm going to talk about biofuel and its global demand. I'm going to talk about the advantages of biofuel and limitations of the biofuel, and how different technologies like proteomics and synthetic biology helps to improve these bioprocesses. So what is biofuel? Biofuel is the fuel derived from the organic matter by sustainable way. So why we are talking about biofuels today? So as we know that the fossil fuels reservoirs or petroleum reservoirs, they are exhausting rapidly nowadays, um, and they are going to be run out one day. We are going to be run out of the fossil fuels one day. It is predicted that by year 2052, we will be completely run out of these stocks, fossil fuel stocks. So you can imagine the life after if you don't have fossil fuels, because most of the energy currently coming from the fossil fuels. So that's the reason. Also, the another reason is fossil fuels has environmental threatening impacts as they generate greenhouse gases, and these greenhouse gases contribute to the global warming. So that is the problem not only uh, for a certain country, it's a problem for whole world. So why this is so? So that's why we need a alternative biofuel which can replace the fossil fuels. Fossil fuels. The global energy demand, on the other hand, increasing by 1.6% every year. Why this is happening? Because of the population growth, and also humans socio-economic status is also uplifting. So, and that's all contributing to the urbanization, and and that's why the global demand energy demand is increasing. So what is the solution? What we need to do? So we need an alternative biofuel which can replace the fossil fuels. The biofuel that that we need a biofuel that will be compatible with the current fossil fuels in terms of energy content. We need to develop the low-cost technology so that the process will be economically feasible. So in short, we need to uh, expand the biofuel production. So we can derive biofuel from different sources based on their substrate. They are classified into different generations. So the first generation of biofuel is the biofuel that we can derive from the food stocks. Food stocks means cereals, grains, and like sweet corn, like that. So the biofuel derived from the uh, food stocks, we called it first generation biofuel, where we ferment the simple sugars like starch into the biofuel. It is found that the first generation biofuel is unsustainable. And why it is unsustainable? Because it is directly competing with the food stocks. So you can imagine, if we use the food stocks to produce biofuel, then what we will eat. So that is the problem, and that's why several international energy agencies, they come out with the idea of the second generation biofuel, because because we have to stop somewhere first generation biofuel. So the second generation biofuel, the biofuel that can be derived from lignocellulosic biomass. Lignocellulosic biomass identify as a prime source of energy, since it is abundantly available in nature. It is renewable and it is cheap, actually, because it is just a waste. We can obtain lignocellulosic biomass in different forms, like forest waste, agriculture waste, even municipal solid waste has an organic fraction that can be converted into biofuel. So it is relatively cheap source of substrate. The most important thing is it is non-food competitive. It is not competing with food stock, so that is good, a good thing. So currently the biofuel market is growing, but most of the biofuel currently produced from the comes from the first generation biofuel. Um, the country like Brazil and America, they produce biofuel from the sugarcane, and mostly it is in the form of ethanol. But as I said, we have to stop somewhere first generation biofuel and start using lignocellulosic biomass for biofuel production. So we have to accelerate this option. Um, so coming to the next slide, as we know that biomass waste, particularly lignocellulosic biomass waste, it is made up of three different kind of polymers. One is lignin, second is cellulose and hemicellulose. So cellulose and hemicellulose, these all polymers, they are interlinked, and they are in a very complex structure. They are present in very complex structure. So 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 the energy, they have tremendous energy, but that energy is trapped in these polymers. Like cellulose is made up of glucose polymers, and hemicellulose is made up of different sugars like xylose and arabinose, different sugars. These all sugars we can convert into fermentable, fermentation byproduct, um, into or value-added byproduct by fermentation. But how we can do that? Because the polymers are very tough. We can't directly convert them into into biofuel. So that's why we need a hydrolysis. That is, we need a pretreatment method that which can break these polymers into simple sugars or fermentable sugars, and that sugars we can utilize for the fermentation. So lignocellulosic biofuel production is based on two main steps. One is hydrolysis and second is fermentation. So the hydrolysis is, we also call it saccharification or pretreatment, which is the bottleneck step in the lignocellulosic biofuel production. Because if we don't break down these polymers, we cannot convert them into value-added products by fermentation. So it is so currently there are several pretreatments the researchers are using uh, to pre-treat this lignocellulosic biomass, particularly physical techniques and chemical techniques. So physicochemical techniques are actually they are energy intensive, because we need a lot of energy input in the process, and that's why the processes are not not economically feasible. And that's why we come up with the idea of biological pretreatment. So in biological pretreatment, we simply use microorganisms or the enzymes derived from microorganisms for the in the pretreatment process, and we hydrolyze these polymers. So biological pretreatment is better than the physicochemical pretreatment, because you can grow microorganism as much as you want. So they are actually renewable, and they are and we can reproduce them, these microorganisms, and we can reproduce the cellulases, the enzymes. So the process based on biological pretreatment is 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 a, is a can be economically feasible. And that's why my research is also focused on improving biological pretreatment of lignocellulosic biomass, and uh, and converting that sugars into biofuel. So biological hydrolysis, basically biological hydrolysis, microorganisms utilizes different cellulases and hemicellulases to break down these polymers. So the advantages of this biological process is, one thing is they are low cost technology. It is not harsh pretreatment, like chemical or physicochemical pretreatments. We don't need chemicals, so it is low cost technology, and it requires very mild environmental conditions. So overall, it is better option than the physicochemical pretreatments. But this method also have the disadvantages. So like it is a very slow process, because the microorganism degrades these polymers in nature very efficiently, but when we bring them into the in vitro condition, they don't work as they work in the same way they work in the nature and natural system. And that's one, we need to improve the hydrolysis process to make it commercially viable. So how we can do that? How we can improve the biological hydrolysis process or biological pretreatment process? We can improve by with the help of synthetic biology. So synthetic biology now comes into play. So what is synthetic biology? Synthetic biology is the transdisciplinary field that intersects with different sciences and engineering field, including biological sciences and chemical engineering. And so synthetic biology have vast applications in different sciences and engineering field. So the role of the synthetic biology is to design or reconstruct the microbial system or biological system with using different new advanced biological parts. So if we're talking about the biological pretreatment, then the new biological parts are actually nothing but the cellulases and hemicellulases. So the objective of the synthetic biology is to provide this biological part to the metabolic engineers to improve the metabolic process. And why we want to improve the metabolic process? To obtain the goal to generate so the goal of the synthetic biology is to generate new chemicals or improve the chemical production. Synthetic biology also has several applications in human health and also environmental issues. So particularly in human health, Synthetic biology plays a very important role in drug discovery, new drugs discovery, diagnostics, and clinical application. It has clinical applications. Also, it plays a very important role in environmental issues like bioremediation, and like waste to biofuel generation is also a part of environmental issues. So overall, synthetic biology is the field which has a diverse application in different sciences. So application of synthetic biology in biofuel generation. What synthetic biology can do in a lignocellulosic biofuel generation? So simply, synthetic biology can develop the robust microbial cells that can produce the vast amount of biofuels from renewable sources by assembling the advanced biological parts or novel biological parts into the biological system. But is it that easy? Engineering of the biological system is particularly difficult because of it is dynamic and it is very complex. It is not a simple like a physical system where the machine, a different part of machine, you can construct them together very easily. And also it is very difficult for synthetic biology to do its function because most of the biological systems are unknown and they are still unidentified, and they are very complex. So we basically lacks the genetic information that requires for synthetic biology to perform its function. So if you see this, this is the cellulosome. I'm just telling you about, giving you one example of the complexity. So cellulosome is actually cellulose degrading system which is present in Clostridium thermophilum, which is a bacteria which degrades cellulose. So this cellulosome is the structure which is present on the cell surface. But if you see, it's not only cellulases present in this complex. There are several non-cellulolytic proteins present in this complex that makes that complex which degrades cellulose. So it has several components, and these several components are fit together and become a cellulosome, a machinery that degrades cellulose. So the complexity, you can imagine the complexity from this structure of the biological system. So basically, we lack the genetic information, we lack the biological understanding of the microbial systems. So here we can get help from a different omics tools like proteomics, transcriptomics, and metabolomics. So what omics does, that they help to identify these biological parts or novel biological parts that can be used for the development of the recombinant microorganisms or robust microbial cells for hydrolysis and biofuel generation. So in my study, I mostly use a proteomics approach to understand the biology of microorganisms and find out these novel biological parts that can help synthetic biology. So the role of proteomics is to identify and characterize these potential targets for synthetic biology to improve the biofuel production. So the proteomics is the field to study the entire proteome of microbial system. The proteins are actually the building block of the cells because they are involved in functions and also in the structure of the cell. So proteomics helps to identify these proteins. In proteomics, so in proteomics, there are several approaches we can use, like different, we can use quantitative proteomics approach. So I used quantitative proteomics approach in my study, where I use iTrack, which is a isobaric tag for relative and absolute quantitation. So what it does actually, it can identify and quantify the proteome of different systems or different cells which are grown in different conditions. It can quantify these proteins in a single experiment. Say, for example, if we have glucose and cellulose grown condition, then iTrack can help to identify the proteome or proteins present in the different samples in a single experiment. And then we can compare that proteome of the different conditions, and we get the relative abundance of the proteins, or relative expression of the proteins in different conditions. So in my study, I use shotgun proteomics approach. Shotgun proteomics approach, as the name indicates, it in this particular approach, we first digest the peptides into proteins into peptides, and then we run them on the mass spec. So the workflow is like this. If you just consider that we have two conditions, these are the microbial cells. So consider these cells are grown in cellulose and these cells are grown in glucose. Once they grow until certain, uh, exponential phase or stationary phase, we harvest these cells, we extract the proteins, and once we extract these proteins, we digest them with the trypsin. The trypsin is the protease enzyme that degrades proteins into peptides by recognizing the arginine or lysine or arginine amino acid, and it cuts the peptide bond after the between arginine or lysine amino acid and the amino acid next to it. It breaks there. So we always get a certain pattern of peptides where the C terminal end is always arginine with arginine or lysine. So once we get that peptides, we run that peptides on the, we further purify them using, liquid chromatography. We use different techniques like strong cation exchange technique or Heilig we use. So based on their ionic strength of the peptides, or based on its hydrophilicity of the peptides, we separate them and we fractionate them. So this is actually, we are removing the unwanted peptides, and we are just collecting the peptides that are derived from trypsin digestion. And once we get that peptide fractionated, we run them on the mass spec, and we acquire the data. We identify the peptides, and then we identify the protein using the databases. So that's the overall workflow of the shotgun or bottom-up proteomics. So coming to the next slide. In this slide, I'm going to tell you how we label these peptides once we digest it with the trypsin. How we label them with the isobaric tags. So what is isobaric tags? As name suggests, isobaric means the mass of they have the same mass basically. So isobaric tags are made up of three parts. One is reporter ion, second is the balance group, you can see here, and reactive group. The reactive group, it interacts with the amino group of the peptides at the N terminal, and sometimes it reacts with the interacts with the side chain of the lysine amino acid in the peptide. So if you see this balance group and the reporter group mass, they are actually, the total mass is 145. So the reporter ions are varying from 117, 116, 115, 114, that are the Dalton. They are they have that mass of 114 Dalton to 117 Dalton. And we have the balance group, which gives, so if you have the reporter ion of 114, then the balance group will be 31. So the total is 145. And similarly, if you have a reporter ion 117, then the balance group is 28. So the total biomass is 145. So like that, all these tags are have the same mass, and that's why we call them isobaric tags. So once we label them, these peptides with the isobaric tags, we get that complex peptide plus isobaric tags. And once we get that labels, so once we label them, we put them together actually in a one sample. So once we label them, now we don't care even if we mix them together, because we already label the peptides from different samples. So once we label, we run them through mass spectrometry. So we get the two spectra, sorry. One is MS spectra and one is MS spectra. So in MS spectra, we get the peak of the peptides, the molecular ions. So molecular ions are actually peptide plus isobaric tags. So we get the peaks for the molecular ions in the MS, in the MS, and in the MS, we get the fragmented pattern of the peptides plus the reporter ions, these reporter ions. So if our peptides derived from different samples are the same, means if they are the identical, the overall mass of the peptide plus tags will be the same. So the MS peak will give you only one peak of all these 4 peptides, because they are identical, so they have the same mass. So they comes at a same time in MS. And in MS, you get this fragmented pattern of the peptide, identified peptide, and the reporter ion. So here, this is the instrument I have used, mass spectrometry. I use Q-exactive hybrid quadrupole orbitrap mass spectrometry. So this is the design of this instrument. So mass spectrometer is typically made up of 3 main parts. One is ionizer, second is analyzer, and third is detector. So once we get that peptides labeled, we fractionate them further with the reverse phase liquid chromatography. We get the different fractions of the peptide samples, and that samples we transfer through the high electric volt electro spray ionization where they get evaporized and ionized, and that ionized molecular ions or peptides, they passes through this quadrupole analyzer system, where basically we select the certain kind of molecular ions or peptides that have based on their mass to charge ratio. And unwanted peptides, they just get destroyed in the quadrupole. And the selected peptides, they transfer to the C-trap, where we collect the certain set of set of the peptides. So say for example, peptides are identical from all 4 samples. So you will get all that 4 amino acids, peptides, the bunch of that peptides in C trap, isn't it? Because they are identical, they will pass at the same time. And once they, once we uh, so once we get that peptides in the C trap, they wait there for some moment, and then they transferred to the orbit trap mass analyzer. So when they reach to the orbit trap mass analyzer, they rotate in the orbital motion around the orbit trap based on their charges and mass and charges, and then they transfer the signal to the detector, and that is how we get the MS spectra. In the similar way, the peptide are passes through this quadrupole, and they collect in the C trap, and during MSMS, this collected molecular ions or peptides with tags, they transfer to the HCD cell, which is collision-induced dissociation, we call it, where the actual fermentation occurs. So we bombard these molecular ions, peptides, or molecular ions or peptides with the inert gas like nitrogen, and we fragment them into pieces. And that pieces again sent back to the C trap, and after from C trap they goes to the orbit trap mass analyzer, and that is how they again do the same thing. They, based on their charge and masses, they rotate around the orbit trap, and they the signals where will be is gets to the detector, and that is how we detect the MSMS, and we get the MSMS spectra. So this is the overall the sketch of the analyzer and the ionization unit of the mass spectrometry. So as I suggest, as I told, you get in MS we get the peak of the molecular ions, which is actually nothing but the peptide plus isobaric tags. And if the peptides are identical, we get only one peak for all peptide derived from all the samples. And in MSMS, the peptides get fragmented, or they broke into the pieces. So and then once they fragmented, the fragments of the peptides, they pass they gives MSMS spectra. So that is how so basically we get two spectra. One is MS and another is MSMS. MSMS spectrum that is actually is the spectra of the amino acids and reporter ions. So here is the fragmentation pattern of the peptide. So if we have the peptide of certain sequence, so suppose this is the peptide which was present in all the samples, all the 4 samples. So we get that we get this MSMS spectra. So then we identify the sequence of the peptides. So how we identify the sequence of the peptide by MSMS spectra? It's very simple. If you see this this Y16 means we get the fragments either the peptide gets fragmented either from N terminal or from C terminal to N terminal, like that. So we got the fragmentation in both directions. So Y16 is here. You can see the Y16 is nothing but the sequence from ESQL. So it's going Y16 to Y15, Y14, or Y13, like that. So if you see the difference between these two peaks from 1959.92 and 1830, the difference between these two peaks is 129 Dalton, and 129 Dalton is actually the ionic mass of the glutamic acid. So that is how we identify. So the most probably the amino acid, this amino acid is glutamic acid. And then if you see the difference between Y14 and Y15 is 87. So 87 is the ionic mass of the serine amino acid. So that is how we identify the complete sequence of the peptides. At the same graph, we get the reporter intensity of the reporter ions as well. So if you see these reporters are 114, 115, 116, and 117, we know that 111 where 111, 114 come from, and where we know that where 115, 116, and 117 come from. So so based on the intensity of this reporter ions, we quantify the proteins. So in my study, I studied the Fibrobacter succinogen, membrane proteomics of the Fibrobacter succinogen at the iTrack based proteomics. So why I did membrane proteome of Fibrobacter succinogen? So first of all, I will talk about the Fibrobacter succinogen. The Fibrobacter succinogen is the microbe. It is anaerobe and it is highly efficient cellulose degrader found in the rumen ecosystem. So Fibrobacter succinogen is very efficiently degrades cellulose, but the problem is that we don't know which mechanism it utilizes. As I told you previously in my previous slide about the cellulose degradation system, this bacteria does not utilize that system for cellulose degradation. So we are expecting that it must have some novel cellulose degradation system, and we probably we can discover in this microbial system, in this cellulose degradation system, we can discover some novel or advanced cellulases and hemicellulases that can help to help synthetic biology to construct or design the most efficient cellulose degrading microorganism. So aim of this study was to characterize and identify key biological parts that are involved in lignocellulose degradation and utilization in Fibrobacter succinogen. So here is the workflow of my study. So basically, I've grown Fibrobacter succinogen in glucose and cellulose condition. The hypothesis behind this experiment is that when we grow Fibrobacter in cellulose on a cellulose substrate, we expect to express the cellulose degradation system or cellulose degradation machinery, and we expect the high abundance of the enzymes and proteins that are involved in cellulose degradation compared to cellulose. So membrane proteomics, as I said earlier, that the membrane proteins are actually, they are very hydrophobic in nature because they are trapped in the cell membrane. So we have to perform the extra step, and that is called as affinity purification step. So we have to purify membrane protein first, and then we can go for mass spectrometry analysis. So in this study, I used biotin neutravectin affinity purification technique. So what biotin does, that it interacts with the amine group of the proteins in the membrane proteins, and they form a complex. That complex, protein biotin complex, then we can enrich with the help of neutravectin, because neutravectin and biotin has affinity, a very strong affinity. So once we got that complex, we can just elute the proteins using reducing agent, and that is how we get the membrane proteins in the solution. So once we got that proteins into the solution, then we go for the angel digestion. And once we did angel digestion, there's we get uh, uh, the peptides, trypsin digested peptides. These peptides then we label with different isobaric tags, as I explained previously. And once we label them, we fractionate them to remove the impurities, uh, and and then we run them on the mass spectrometry, and we get MS and MS in this data. So like that. So with this study, what we found that we quantified a different, large number of membrane proteins that are regulated or that are abundantly found in cellulose grown cells. And based on that data, we propose the mechanism of cellulose degradation in Fibrobacter succination. So you can see that there are so many things happens when they degrade cellulose compared to glucose grown cells. So we found that there was number of proteins were upregulated or they were found in high abundance in cellulose grown cells. So we found that there is a novel secretion system which is involved in cellulose degradation, which is which is which is first time we saw in which we first time saw that involved in cellulose degradation. So that's T9SS secretion system. What it does, that it brings all the cellulases and hemicellulases on the cell surface of the bacteria. Then it also brings exports the adhesion proteins there, which are not actually cellulose degraders, but they are helping to attach the cells to the cellulose particles, like here fibroslime protein. So like that, they come together and they that machinery, and they build that cellulose degradation machinery on the cell surface. And then cellulases come very close proximity to the substrate like cellulose, and they degrade the cellulose polymers into pieces. And these pieces are actually, they can be glucose or cellobios or cellotriose or cellotetros. These are all byproducts then sent back to the periplasmic membrane by beta barrel, which is one kind of transporter. Once we got this cellulose degraded byproducts in the periplasm, they are further degraded by other enzymes, and they are transported into the cytoplasm using ABC transporters. That is how the bacteria gets glucose and cellobios for its growth, and that is how it utilizes this sugar in its metabolism. So we found several parts, several like CDMs, GH, that is glycosyl hydrolases, means they are cellulases. So cellulases and cellulose binding modules, these are all novel proteins. Even T9SS secretion system, these are all novel parts we identified in these cells. And we also found that there are proteins which are involved in cyclic di-GMP production. These the cyclic di-GMP production, this particular secondary messenger is actually involved in biofilm formation. So we found a very dominant biofilm formed by the Fibrobacter succination when it grows on the cellulose particles. So this is the overall mechanism we obtained from by this study. We published our study in scientific report recently. You can have a look on that paper for more details. Um, so these are all the parts what we have identified or quantified by the proteomics method, which are involved in lignocellulose degradation. They are all are the novel biological parts. So these parts now we can give it to synthetic biology to help to construct or design the recombinant organism, a robust recombinant organism that can rapidly degrade lignocellulose, and then we can ferment it into the biofuel. So that is that is how proteomics helps to understand the biology of the microorganisms and helps to identify the biological parts like enzymes and the different proteins that are involved that have the desirable functions. So that's all from my talk. I would like to thank my colleagues, my supervisors, and thank you for your attention.